Hi, my name is Stephanie Varghese, and I'm an undergraduate researcher of plant ecology in the Wright Lab here at Cal State LA. I am extremely grateful for the opportunity to share with you today a glimpse into the research on our novel dehumidification strategy for enhancing plant drought experiments. Global temperatures are rising, and so is the frequency and severity of Mediterranean drought. Contemporary plant drought experiments aim to simulate natural drought by reducing soil moisture via controlled watering regimes and rainout shelters, two of which are shown here on the right. But a key component that is oftentimes overlooked is atmospheric moisture, because up until now, we haven't necessarily had a practical, low-cost, and effective means of simulating atmospheric drought. Our lab came up with a novel solution, which we found to work highly effectively in our drought experiment on campus, which is located just outside the Annenberg Science Complex. Our novel dehumidification treatment involves silica gel packets. Silica gel itself is a solid granular form of silicon dioxide with a massive internal lattice of interconnecting microscopic pores. This material is capable of adsorbing water vapor from the environment and then retaining that moisture through a process known as capillary condensation. Packets continue to desiccate in this way until an equilibrium state is reached between the moisture level in the packets and the moisture level in the surrounding air. But because this is a passive process driven by concentration gradients, water molecules can travel in either of two directions: into a dry packet on a humid day, or out of a saturated packet on a dry day. We wanted to understand the different factors that might activate that adverse humidification effect we see on the right. Likewise, our first hypothesis is that the degree of daytime drying by silica gel packets is affected by air humidity, soil moisture, and the frequency of packet replacement. This first figure shows the effects of the macroclimate relative humidity on the desiccation effect. The desiccation effect is shown here on the y-axis. The lower the value is, the stronger the dehumidification effect. We found that packets work highly effectively on rainy days when we have a rainout shelter in place, as indicated by these X-shaped data points, and also on humid days in general. We also found that packets dehumidify the air effectively when combined with a watering treatment designed to reduce soil moisture. Packets might be working less effectively in pots of higher soil moisture because they might increase the rate of evaporation off the soil surface, saturate with soil moisture, and then have less space to accommodate for air moisture. But we did find that packets work highly effectively in our pots of drought watering, as is the case in contemporary plant drought experiments. We also suspected that packets' desiccation ability might be damaged by leaving them out for several days on end. We found that packets work effectively for the first 3 to 4 days and then need to be replaced thereafter. Our second hypothesis is that the nighttime dehumidification effects in California are stronger than the daytime effects. Our results supported this hypothesis, likely because California's climate consists of hot, dry days and cool, humid nights. During nighttime, there might be a higher content of moisture for packets to adsorb, which might explain why the dehumidification effect is stronger at nighttime. Our third hypothesis is that the degree of dehumidification depends upon fine-scale changes over the course of a 24-hour day. This next figure illustrates the 24-hour dehumidification effect on an hourly time scale. We separated this figure into 3 distinct panels, so it's easy to see how the dehumidification effect of silica gel packets might vary in climates characterized by low, medium, and high relative humidity. We see here that packets respond very quickly and throughout the 24-hour day to changes in air humidity. In other words, there doesn't seem to be much evidence suggesting that packets require a whole lot of time to adapt to changes in air relative humidity. Hopefully this next conceptual figure helps clarify that last point. Overnight, packets reach the highest point of saturation. By 8 a.m., packets may emit the previous night's humidity when air moisture begins to drop. By 9 a.m., packets may have emitted enough moisture to work as effective dehumidifiers again. But as air moisture continues to drop at 10 a.m., and then throughout the remainder of the day, packets may either adsorb or emit moisture, depending on the moisture content on either side of the air packet equilibrium. So to summarize these findings, we discovered that silica gel packets work most effectively when they contain a lower moisture content than the surrounding air. This can be the case when the macroclimate is humid, when it is nighttime in California's climate, and when a rainout shelter is in place. Similarly, we found that packets are more likely to counterproductively humidify the air when they contain a higher moisture content than the surrounding air. This can be the case when packets saturate with soil moisture and when they haven't been replaced in several days. And finally, we found that packets can enhance present-day drought studies by absorbing excess humidity produced by rainout shelters, which is one of the primary soil moisture manipulations, and reducing air humidity to drought levels on particularly wet days, which is when the atmospheric conditions are least similar to what we would want to simulate in a drought experiment, and therefore when an effective dehumidification treatment is most needed. Here are my references. And with that, I would like to thank my advisor, Dr. Wright, my thesis committee, the Moore programs of Cal State LA, and everyone in the Wright Lab who helped to make this project possible. I will now take any questions.